We are continuing today with carbohydrate metabolism. And if you look at the course outline, as we always do, we look at structure, function of the biomolecule, then we look at catabolism, and then we go ahead to look at anabolism. Today we are going to take a look at one of the catabolic processes of carbohydrates, which is glycolysis. So the main metabolic process of carbohydrate we'll look at today is glycolysis. But we'll look at some other aspects in addition to glycolysis. Now, generally, we have known carbohydrates to be the component of food that provides us energy. We've known that from primary school all the way to the university. We've always talked about carbohydrates as a source of energy. And when you look at meals that we take globally across the world, you will see that carbohydrate forms a major component of our meals. So, for instance, if you take Ghana, for instance, the major foods, the staple foods that we eat, banku with some soup or some sauce, and so the major part of the food there is carbohydrates. If you take fufu, the same, fufu with some soup. If you take rice, you see rice with some stew or some soup. So the rice is forming a major component there as a source of carbohydrates. Or even if you are taking in some bread with some tea, you see that the bread is a major source of carbohydrate there in that breakfast meal. And if you go across the world, you realize that carbohydrate forms that major component in different ways, in different forms. So, for instance, if you are in Europe, you find that a lot of their meals will be some soup with some bread, different kinds of breads that they will take. It's just like in place of our banku and fufu, they have some different types of breads that they'll be taking with some vegetable soups. You go to Asia, they will take a lot of these cereals like rice. Anyway, Ghana now, rice is also eaten every day, just like the Asians. So yes, you find major source of food being carbohydrates. If you go to northern Ghana, a lot of the carbohydrate source we have there will be the yam and millet, sorghum, guinea corn. You'll find those things there. So actually what happens is, if you take plants which are photosynthetic, they are able to produce their own carbohydrates by the process of photosynthesis. And you can refer to photosynthesis as a method by which plants prepare their own foods. And what are the ingredients that the plant uses? The plant uses carbon dioxide and water. And you see, if you say carbohydrates, we saw the structure of carbohydrates. Carbohydrates. So it's carbon and water. And you see, when you take the molecule carbon dioxide and water. I mean, the elements you find in there, carbon, hydrogen, oxygen. That's what you find in a carbohydrate too. So the ingredients, carbon dioxide, water that a plant is using, and uses energy from the sun. So it's using some light energy there to prepare the food, the carbohydrate. And when it prepares the food, it stores the food in its roots as starch. So you see, plants store carbohydrates as starch. They store it as seeds. That is when you go in for and then grains. So if you go for maize, sorghum, guinea corn, rice, that's carbohydrate starch that is stored by the plant in those parts of the plant. Root tubers like cassava. So the plant is storing carbohydrates in the roots. Or maybe you take sugar cane, for instance. That is carbohydrates, the disaccharide, sucrose, the common sugar, which the plant is storing in the stem. There are some stem tubers like yam, like potatoes, that you see that, yes, the plant again is storing starch in those parts of the of its structure. And so for animals, say for us mammals like humans, that we are not photosynthetic, we cannot prepare the carbohydrates ourselves. So we go in for these carbohydrates from the plants. We take the root like the cassava. We take the grains like the maize. We even go for the fruits that contain other sugars like sucrose, fructose. We eat those fruits. So we get the carbohydrates from those parts of the plant that the plant has stored the carbohydrate that it has prepared. Then, because when we take in these carbohydrates, we break it down, say to simple sugars, and we are not able to consume all of it at once. I mean, if you take carbohydrates and you break it down to glucose, and you have blood glucose, the body does not make use of all of it at once. It stores some of it. So some of these carbohydrates we store as glycogen. So as the plant is storing as starch, we store as glycogen. And we store the glycogen in the liver. We store some in the skeletal muscles. When we are sleeping, the glycogen breaks down gradually into glucose into the bloodstream, and we make use of that. And so we wake up in the morning and then we break our fast. Or when we are involved in some vigorous activity that the muscle cells need energy so quickly, we break down the glycogen in the skeletal muscles into glucose molecules to generate energy for the body. Yes, patience. I saw that the screen went off, even my voice went off, the connection cut for a short period of time. So I stopped talking and waited. I hope now you can see it and you can hear me. Okay, so from that introduction, we're looking at the outline that we should be, covering over the period that we are studying metabolism of carbohydrates. And these are the 5 metabolic pathways, the major metabolic pathways that we should be looking at over a period. Glycolysis, TCA cycle, glycogenesis, glycogenolysis, and the pentose phosphate pathway. Today we're looking at glycolysis, and then some other 2 pathways in addition to glycolysis. TCA cycle, glycogenesis, glycogenolysis, and the pentose phosphate pathway. We'll look at them together in our next lecture. So if you see, what we'll do is that if you look at the pathway on the right-hand side, you see glucose going down to pyruvate. So that process is what is referred to as glycolysis. Glucose undergoing some reactions all the way to form pyruvate. That is glycolysis. And that pyruvate also undergoes some reaction before it goes into a cycle, what we refer to as a citric acid cycle, which is the same as the tricarboxylic acid cycle. That's what we are referring to as the TCA cycle. That we will take in the next lecture. The continuation, which is oxidative phosphorylation, that's going to be happening in the mitochondrion, which is what we also refer to as electron transport chain. That one is, we are not going to do that. We'll end it at the citric acid cycle when we are taking the TCA cycle and the other pathways in the next lecture. But today we'll take glycolysis, and then we'll also look at carbohydrate metabolism in anaerobic conditions. So we'll look at some fermentation processes in addition to that process of glycolysis. That's going to be for today. So when you take glucose, so glucose as the simple sugar, these are the 3 major pathways that glucose can undergo. Of course, even if you look at the slide we're looking at above, there are other pathways there, but the major pathways, first of all, before you have other pathways connected to it, are what we are showing here. We see the process of glycolysis. So we have oxidation via glycolysis, which produces pyruvate. So that is the major process we are going to look at today. So that is catabolism. And we have to the left, you have also another oxidation process, which we refer to as a pentose phosphate pathway. This goes into the production of ribose 5-phosphate. And that ribose 5-phosphate will go into the synthesis of nucleotides. Because if you remember looking at the structure of nucleotides, remember that you have the ribose 5-phosphate on which you have a nitrogenous base. That gives you a nucleotide. And those nucleotides then go into the synthesis of nucleic acids, RNA, because this is ribose 5-phosphate. Those will go into synthesis of RNA. If this sugar undergoes some say dehydroxylation, what you have is that you now have a deoxyribose 5-phosphate, and that will go into the synthesis of DNA. So, and even you have some of these nucleotides going into the synthesis of coenzymes, like some nicotinamide dinucleotides, flavin dinucleotides. Those are some coenzymes that you have those nucleotides going into. So believer, Akoli says, I can't see your screen, sir. Well, I'm monitoring it from another device. I see everything is showing. So perhaps your network must be having an issue there. Usually I see some of your friends, anytime this thing occurs, they say you should go out of the class and come back and it will show. So maybe you want to try that and then it will be visible for you. So these two pathways downwards are like catabolic pathways. But the pathway which is upwards is an anabolic pathway. I mean, that is where you have glucose going into the synthesis of starch in plants. Sucrose also in plants like sugarcane or maybe sugar beets. And you have glycogen in animals, what we talked of, the storage in the liver and the skeletal muscles. So these are the major pathways that glucose undergo. Now we can go ahead and start to look at the process of glycolysis. What we mean by that, glycolysis. So even from the name, the term, glyco means sugar. Then lysis means breakdown. So glycolysis simply means breakdown of sugar. Now, glycolysis is broken down into two phases, phase 1 and phase 2. Each of these phases consists of 5 reactions. So the first phase, the phase 1, sometimes is also referred to as an investment phase or preparatory phase. And phase 2 is also sometimes referred to as the payout phase. I mean, if phase 1 is investment phase, then phase 2 should be a payout phase. And we'll come to understand why such a term, investment phase and payout phase. Now, what we'll do is that we will take each of the reactions one after the other. You know what I say, if you have a complex reaction pathway, you don't get intimidated and scared about it. You know how to write chemical reactions. Reactant going to product. One arrow at a time. So let's start. First arrow. Arrow number 1. Glucose goes to glucose 6-phosphate. And we have ATP in that reaction. ATP gives out 1 phosphate group onto glucose. That's why you have a glucose now onto the carbon number 6 becoming glucose 6-phosphate. And when ATP gives out 1 phosphate group, it's left with 2 phosphate groups. So you now have ADP, adenosine diphosphate. So from adenosine triphosphate, you have adenosine diphosphate. Now, an interesting thing about this reaction is, I think I've mentioned it before, that one of the ways of activating molecules to go down a reaction pathway is by phosphorylation. So you see, you've been referring to ATP as a form of energy. Yes, it's a cellular form of energy, and this is how the energy is used. It phosphorylates molecules, and that will activate the molecule to go down a reaction pathway. And so you see, beside it, it's written first priming reaction. That's activation. Now, what you see is that on the side here, where you have the structures drawn, you see each of the structures that are in the reaction, you have each of those molecules in the reaction, you have the structures drawn beside them so that you can easily relate. So glucose, I mean, you have seen glucose molecules from first year. And glucose 6-phosphate, you see that on that carbon number 6, where there's a hydroxyl group, in place of that, you now have the phosphate group. That's a phosphate that came from ATP. Now, you see the arrows are numbered. You have number 1 there. And that number 1 corresponds to an enzyme. So you see the enzyme also written at the right-hand side, which is hexokinase. And we have talked about enzymes. I think when we were doing proteins, we looked at a major function of proteins as enzymes, and we did some enzyme classification. So if you see the name kinase, I mentioned that kinases are enzymes that are involved in the transfer of phosphate group from one molecule to the other. And here you see there's a phosphate group that is being transferred from ATP onto glucose to give you glucose 6-phosphate. And the substrate there, glucose, is a hexose. So you have that enzyme, hexokinase, catalyzing that reaction. Arrow number 2. Glucose 6-phosphate goes to fructose 6-phosphate. That's an isomerization reaction. I mean, glucose and fructose are isomers. So it's just some rearrangement of the atoms in the molecule to give you that fructose 6-phosphate from glucose 6-phosphate. And so you see, from a 6-sided ring, that's a furanose, you are now having a paranose. And the enzyme there, phosphohexose isomerase. So the name of the enzyme here even tells you isomerase. That reaction is an isomerization reaction. Arrow number 3. Fructose 6-phosphate goes to fructose 1,6-biphosphate. And we see ATP coming in that reaction again to give out a phosphate group. That's why you see that there was just one phosphate group, fructose 6-phosphate. Now it's a fructose 1,6-biphosphate, biphosphate. So 2 phosphate groups. And so you see, if you want to look at the structure of fructose 6-phosphate, there's only one phosphate group on carbon number 6 here. But when you look at the structure of fructose 1,6-biphosphate, you see there's a second phosphate group on carbon number 1. So you now have a biphosphate. And the enzyme which is responsible in that reaction is phosphofructokinase 1. So that's another kinase. This time around is acting on a phosphorylated fructose. That is fructose 6-phosphate. And so you see the enzyme is phosphofructose kinase 1. Now, you'll notice that this fructose 1,6-biphosphate, you have it partly shaded. You have a portion of it which is shaded and a portion unshaded. Why that? Now pay attention. In this reaction number 4, this is where we are going to see the lytic reaction, the breakdown, the lysis. In the name, glycolysis. So the lysis is coming to occur in reaction number 4. And this shaded part is what is becoming this molecule that is shaded. And the unshaded part is the second molecule, which is unshaded. Now, you take a look at that reaction, and you see you have 2 molecules produced. The first molecule, which is shaded, is glyceraldehyde 3-phosphate. And the second molecule, unshaded, is dihydroxyacetone phosphate. So you have 2 molecules now from 1 molecule of glucose. The glucose is broken down. Glucose contains 6 carbons. Now the molecule is broken into 2. Each of these molecules contains 3 carbons. So you see, the glyceraldehyde 3-phosphate contains 3 carbons. Dihydroxyacetone phosphate also 3 carbons. So the molecule there is broken down into 2. And we see the enzyme responsible in that process is an aldolase. Why an aldolase? I mean, that's because you have an aldehyde formed, glyceraldehyde 3-phosphate. That's the major, product that is formed there. That's why it's being referred to as an aldolase. But a fit reaction is just a conversion of that dihydroxyacetone to a second molecule of glyceraldehyde 3-phosphate. So what it means is that in the first phase of glycolysis, you have glucose broken down to 2 molecules of glyceraldehyde 3-phosphate. And so you see the last name below there, glyceraldehyde 3-phosphate, is having into bracket 2, meaning you have 2 molecules now. From 1 molecule of glucose, you now have 2 molecules of glyceraldehyde 3-phosphate. And it is these 2 molecules of glyceraldehyde 3-phosphate that will go into the second phase of glycolysis. The enzyme responsible there for the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate is also an isomerase, triose phosphate isomerase. It is converting a triose. The sugar is a triose, and that sugar has a phosphate group on it. It's dihydroxyacetone phosphate. I would say triose phosphate isomerase. Now, what you should also pay attention to is the fact that, you see, the breakdown of carbohydrates, we always refer to it as to generate energy. Not so. Yes. The whole purpose of the breakdown of this glucose molecule is to generate energy for us. But here is the case in reaction number 1 and reaction number 3, energy is rather being used. ATP is a form of cellular energy. The process is supposed to generate energy, yet in this first phase, energy is being used. That's why it's being referred to as an investment phase. So we'll see if the investment of these two molecules of ATP energy is profitable or not. Because in the end, the process must generate some energy for us. And we'll see if the energy it generates is more than what is consumed or not. Now, all these that we have just gone over, this first phase of glycolysis, we'll go over the phase 2 of glycolysis as well. And I know by now your mind is also telling you, so how is exam going to be like on this sort of thing? Because I know how sometimes students, when they see some reaction pathways like this, they are wondering in their minds, are we going to be asked to draw all of these reaction pathway? Are we going to be asked to draw these structures? Well, if you were maybe doing a biochemistry major, yes, you will face examinations where you produce this and you give some explanations and things like that. In your case, you are doing a HND, Science Laboratory Technology, and you are doing biochemistry as a course. So questions that can come from these, for instance, can be questions about the enzymes, because we did study enzymes prior to coming to this. So to test your understanding, you can ask question about classification of the enzymes. So for instance, if I give you the reaction pathway and you look at glucose going to glucose 6-phosphates, and you know the enzyme is hexokinase, can ask you in which class will you put this enzyme, for instance. And you should understand that what is happening here is a transfer of a phosphate group. And even in the name, kinase, it tells you that. And we looked at 6 classes of enzymes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So looking at the reactions that are occurring, you should be able to classify the enzymes. I mean, that should be simple. If you really understand, you should be able to classify these enzymes. Or maybe sometimes questions can be asked about what type of reaction is it that is occurring. And that too, you can also determine it even from the name of the enzymes, or just by looking at the structures. You should be able to tell whether it's an isomerization reaction, for instance. You should be able to tell, or phosphorylation reaction. I mean, this is simple. You can see that phosphate group is being transferred from ATP onto a molecule, and so that is a phosphorylation reaction. Or even this reaction number 4, that you are having a breakdown of the glucose molecule. What type of reaction is that? It's a hydrolytic reaction. Even though you don't have water showing there, it's a hydrolytic reaction because it's a breakdown of one molecule into 2. That's where the lytic reaction is occurring. So those are some of the questions that can come from these things. In any case, I think I've given you self-assessment questions for the first part. So when we finish these two on carbohydrates, sure, you have self-assessment questions. Right. Now, one interesting thing that we can talk about, even before we go on to the second phase, relating these, in practical terms, this is particularly in clinical settings where you have some tests that are based on this reaction metabolic pathway that you have, where you have the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. And if you study that reaction more in detail, you see the conversion of dihydroxyacetone phosphate goes to an intermediate, which is inidiol, before getting converted to glyceraldehyde 3-phosphate. I mean, that reaction is fast. It's fast. But if there is an elevated amount of glucose in blood, that is in some diabetic conditions, even in some cancerous conditions, what happens is that you have elevated amounts of these intermediate inidiol, which then goes into the formation of methyl glyoxal. Even though that pathway is slow, once you have elevated amounts of glucose in diabetic conditions, that you have some of these dihydroxyacetone phosphate going into the formation of methyl glyoxal, you will be having an increase in methyl glyoxal. So methyl glyoxal will be above a certain threshold in blood, and that becomes a biomarker. You have some glyoxal tests that is used for diagnosing diabetic conditions. This methyl glyoxal easily forms, easily binds to blood proteins like albumin, hemoglobin. So you have glycated hemoglobin. These days, you have some of these blood tests, which we refer to as glycated hemoglobin tests, to show that you have elevated amounts of glucose in blood, to diagnose some diabetic conditions. So methyl glyoxal there is used as a biomarker for that. Okay. Now, to phase 2. Remember, in phase 1, we had 2 molecules of glyceraldehyde 3-phosphate at the end in phase 2. And those 2 molecules now go into the formation of 1,3-biphosphoglyserates. And that is just a form of oxidation reaction. And you have both phosphorylation and oxidation occurring in that process, because you are having an inorganic phosphate. This time around, it's not ATP. It's just inorganic phosphate groups that are binding on carbon number 1. So that if you look at the structure from the right, you see glyceraldehyde 3-phosphate is having 1 phosphate group on this, carbon number 3. But the biphosphate which is formed now has a second phosphate group on carbon number 3. on carbon number 1. And so you now have 1,3-biphosphoglycerate. And pay attention that all these molecules are 2,2,2 molecules. So you see beside them into bracket 2. Because we have 2 molecules of glyceraldehyde 3-phosphate from the phase 1. And you also see this coenzyme. I think when we were doing enzymes, we did look at coenzymes. NAD+, it's a common coenzyme we've seen to be involved in oxidation reduction reactions. You see there? So there is oxidation occurring. Itself, it accepts a hydride and becomes reduced, which means the substrate, glyceraldehyde 3-phosphate, becomes oxidized. So from an aldehyde, you see, if you look at this aldehyde group, C double bond O, H, now that is having a second oxygen there, C double bond OO, a phosphate group. What enzyme is catalyzing the reaction? Glyceraldehyde 3-phosphate dehydrogenase. And we have seen that dehydrogenases are oxidoreductases. We even use the word interchangeably. Oxidoreductase, dehydrogenase, we use it interchangeably. So quickly, that tells you that enzyme is involved in an oxidation reduction reaction. Reaction number 7. 1,3-biphosphoglycerate goes to 3-phosphoglycerate. And here we see that the phosphate group that is lost from the first position, because from 1,3-biphosphoglycerate you have 3-phosphoglycerate, which means the phosphate group on carbon number 1, that first position, has been transferred. Where is it transferred to? It's transferred onto adenosine diphosphate, ADP, to give you ATP. Oh, this is interesting. You remember that in the first phase, we used 2 molecules of ATP, and we said that was an investment. 1 molecule we used was in reaction number 1, and the second molecule was in reaction number 3. And now we are producing 2 molecules of ATP. Okay, so that means that in this reaction number 7, we have paid for that ATP, which we have used already in reaction in the first phase. So of course, if after this, we see a production of some ATP again, that obviously must be a profit. Well, even though there's some energy which has been produced already, which we'll come to later, we'll add all of it together and see if the glycolytic pathway is profitable or not. And the enzyme there is phosphoglycerate kinase. Kinase. Again, an enzyme involved in the transfer of a phosphate group. This time the phosphate group is from 1,3-biphosphoglycerate onto ADP to produce ATP. Reaction number 8. 3 phosphoglycerate goes to 2-phosphoglycerate. I mean, this clearly tells you that this is an isomerization reaction. The phosphate group has just moved from the third carbon to the second carbon. And you see it there in the structures that you have the phosphate group on carbon number 3, and now you see the phosphate group in the carbon, on the carbon in the middle, carbon number 2. So that's an isomerization reaction. And we see even the name of the enzyme says phosphoglycerate mutase. I mean, this time around, it didn't say isomerase, but it says mutase, which is the same thing. It's just like, from the word mutation. You can actually put in place of that mutase isomerase. So you can say phosphoglycerate isomerase. It's the same. We go to reaction number 9. What do you see? There is removal of water molecule. And when that happens, you have phosphoenol pyruvate. And what do you notice? You notice that from 2-phosphoglycerate, when the water molecule is removed, so you have some groups in that molecule removed. It's not that the molecule is broken down into 2, but you have some groups removed from it. A hydroxyl group and a hydrogen. And it's giving you water. And when that occurs, you have a double bond formed. Double bond is formed. So you have this hydroxyl group on carbon number 3 removed, and the hydrogen on carbon number 2 removed. And you now have a double bond between carbon number 2 and carbon number 3. Which class of enzymes carry out such reactions? Lyases. Lyases remove groups from molecules or add groups to molecules. If it removes groups from molecules, it results in the formation of double bonds. If it adds groups to the molecule, it removes the double bond. So the double bond. So if a reverse of this reaction, see the reaction is a reversible reaction. If that there's water rather added to phosphoenol pyruvate to give you the 2-phosphoglycerate, then what will happen is that you have the double bond removed, and you will now be having a single bond. So lyases, and you see the enzyme there is enolase, coming from what is produced. It's a phosphoenol pyruvate. So you say an enolase. That enolase is a lyase. Then the last reaction. Interesting already. I'm seeing here that there is production of 2 molecules of ATP. That certainly must be a profit because we've paid for that ATP which we used already in phase 1. So this certainly must be a profit. 2 molecules of ATP produced. And this time too is a transfer of the phosphate group in phosphoenol pyruvate onto ADP to give you ATP. The product, pyruvate. That is the end product of glycolysis. From 1 molecule of glucose, you have 2 molecules of pyruvate in the process of glycolysis. The enzyme acting in that last reaction, number 10, is pyruvate kinase. Again, kinase, transfer of phosphate group. is occurring. So the enzyme there is a kinase. If you are classifying it, it's a transferase. So all those kinases we've been seeing, they are all transferases. They're involved in transferring a group from one molecule to the other. So they are transferases. All right. Now, apart from these two molecules of ATP that are produced in reaction number 10, in reaction number 6, we see there is a production of two molecules of NADH. Now, what you must know is that NADH is also a form of cellular energy. One molecule of NADH is equivalent to 3 molecules of ATP. So what it means is that if we have 2 molecules of NADH, that is equivalent to 6 molecules of ATP. So certainly, the process of glycolysis is profitable. In phase 1, 2 molecules of ATP were used. In phase 2, you have, if you put all together, the 2 NADH there gives you 6 molecules of ATP equivalent, plus another 2, which is in reaction number 7 formed, that is 8 molecules of ATP, plus the one in reaction number 10, that will give you a total of 10 molecules of ATP produced in phase 2. So if you subtract what has been used, the investment in phase 1, you have a profit of 8 molecules of ATP. We can take a look at that calculation much more later. Now, another practical thing, also clinically, that we can take a look at in the conversion of 1,3-biphosphoglycerate, when you have one of the phosphate groups going into the formation of ATP. That is in reaction number 7. So this is where we are talking about. You have 1,3-biphosphoglycerate giving out the phosphate group on carbon number 1 onto ADP to form ATP. And the product there is 3-phosphoglycerate. So we are zooming in on this reaction number 7. All right. So this is a reaction. 1,3-biphosphoglycerate going to 3-phosphoglycerate. And the enzyme there is phosphoglycerate kinase. Now here too, though this reaction pathway where you have the 3-phosphoglycerate, the next thing is isomerization, where the phosphate group moves from the carbon number 3 to carbon number 2 to give you the 2-phosphoglycerate, where we saw that enzyme phosphoglycerate mutase catalyzing that reaction. Now, this reaction occurs, in all cells. You have this reaction occurring in all cells, so that even you can have reconversion, conversion of this 3-phosphoglycerate to 2,3, which means phosphorylation can occur to that 3-phosphoglycerate on, say, carbon number 2. So you'll be having 2,3-biphosphoglycerates. Or even when we have that 2-phosphoglycerates, there can be phosphorylation on carbon number 3 for us to get 2,3-biphosphoglycerates. So that occurs in all cells, at low levels. And something specific occurs in red blood cells, also in placental cells, where the 1,3-biphosphoglycerate gets converted to 2,3-biphosphoglycerate. So the same 2,3-biphosphoglycerate, or from the 3-phosphoglycerate, you have phosphorylation for us to get 2,3-biphosphoglycerate. This occurs in red blood cells and placental cells. Why in red blood cells and placental cells? All right, so this 2,3-biphosphoglycerate we refer to as an allosteric effector. If you are studying the function of hemoglobin that is carrying oxygen from the lungs to the tissues, and then carbon dioxide from the tissues back to the lungs to be exhaled out of the body, we have some chemical entities that we refer to as allosteric effectors in the tissues. And what do these allosteric effectors do? They facilitate the release of oxygen from the hemoglobin. When it brings the oxygen into the tissues, these chemical entities help the release. And one of such is the 2,3-biphosphoglycerate. This sort of reduces the affinity of hemoglobin for oxygen. And that, of course, will cause hemoglobin to release the oxygen molecules fast to the tissues and pick the carbon dioxide, which is produced from cellular metabolism, back to the lungs to be released. And, you have the placental cells also involved in, moving oxygen to the fetus. And so you have a lot of these 2,3-biphosphoglycerate also in the placenta, helping in the release of oxygen to the fetus. And even much more practically, even say in sports, you hear that sometimes when you have athletes going to have some big tournament, they go to some high altitude to train. So for instance, you hear say Ghana Black Stars are going to Kenya to go and train in the mountains. And that is because, the biochemical foundation for that is what we are looking at here, that at a higher altitude, you have less atmospheric pressure. And at such higher altitudes, you have lesser amount of oxygen. And in that condition is some form of a hypoxic condition which is created. And in that condition, you have the body producing more of these 2,3-biphosphoglycerates, which is an allosteric effector, which will cause hemoglobin to be releasing oxygen molecules faster to the tissues. So at that altitude, when an athlete goes to train there for some time and gets adapted to that condition, when he comes down from that altitude to a lower altitude where atmospheric pressure is higher and more oxygen is present, the performance of that athlete will be higher because there will be more oxygen now present at the lower altitude, whilst the release of oxygen from the hemoglobin will be much faster. And so the performance will be higher. So you see, biochemistry is not just some abstract thing looking at chemical processes in biological systems, but this is where you see biochemistry also being applied, even in sports. Right. So the calculation that we were looking at much earlier, we can look at it much more in detail, where we say in the first phase, 2 molecules of ATP was used. Then, in phase 2, 2 molecules of NADH was produced plus 4 molecules of ATP. That's in phase 2. And we also saw that 1 molecule of NADH is equivalent to 3 molecules of ATP. So, if you're calculating the net energy that is generated in the process of glycolysis, where we subtract the amount of ATP equivalent that is produced from, we subtract the amount of ATP that is consumed from the ATP that is produced, we see the net ATP equivalent. 10 molecules of ATP, we calculated that to mean that first, in that second phase, you have these 3 NADH being equivalent to 3 molecules of ATP times 2, that is 6 molecules of ATP equivalent plus 4 becomes 10. So that 10 molecules of ATP minus the 2 molecules which were used in the first phase. And so the net ATP equivalent produced in glycolysis is 8 molecules of ATP. Sometimes you are asked, what's the gross energy produced in the process of glycolysis in terms of ATP equivalent? That, of course, will be 10. That is, if you are not subtracting what has been used from the first phase. That's gross. But the net, it means you are subtracting what you used from what you produced. You have 8 molecules of ATP there. Right, so that's a simple mathematics there that you can do to quantitatively look at the amount of energy which is produced in the process of glycolysis. All right, now, I also said we'll be looking at some 2 other processes in anaerobic conditions in addition to the glycolysis. So after glycolysis gives us 2 molecules of pyruvate, this pyruvate can undergo different pathways. We have 2 pathways being shown here, which are anaerobic. One will go into the formation of ethanol, and the other will go into the formation of lactate. The central pathway is what we'll be looking at in the next lecture, in order to be under aerobic conditions, going into the TCA cycle. That's what we'll be looking at. We can look at it much more here, that we have glycolysis producing pyruvate. So what you should know is glycolysis, occurs, it's not dependent on whether it's anaerobic or anaerobic condition. It occurs at all times. But after you have pyruvate, if you have an aerobic condition, that is, if you have oxygen present, that pyruvate is converted to acetyl-CoA. And then that acetyl-CoA goes to the TCA cycle. So that central pathway is what I'm saying we'll be looking at in our next lecture. Now the reaction pathway, which is going to the top, will be occurring in animals. under anaerobic conditions. That is when oxygen is absent. That's where you see the enzyme lactate dehydrogenase converting pyruvate to lactate. The reaction pathway which goes down will be occurring in bacteria and yeast. That goes into the formation of ethanol. So the first reaction you see, there's a conversion of pyruvate to acetaldehyde. And the enzyme there is pyruvate decarboxylase. The next reaction, alcohol dehydrogenase, acts on acetaldehyde to give you ethanol. Now, we can take a look at these two processes. Let's zoom in on each of them and take a look at them much better. Now, let's just take it from pyruvate. Don't bother yourself about the top there because we've looked at all this already. Glyceraldehyde 3-phosphate going to 1,3-biphosphoglycerate. That we have seen already, but we take it from pyruvate. So pyruvate is here. So you see, we are looking at the ethanol fermentation reaction. It is this reaction which is applied in the production of ethanol, even in the food and beverage industry. In the breweries, yes, they make use of yeast for the production of ethanol. And in some industrial processes where there is production of enzymes to cause some fermentation processes, yes, using some bacteria. This reaction is employed in those reactants, reaction digesters that are used in the industries. Now we see pyruvate. First thing, there's the process of decarboxylation. So you see there's a carbon dioxide removed here. Just take it like this carboxylic acid group is what is being removed to give you that carbon dioxide. And you are left with acetaldehyde. It's an acetaldehyde. And that acetaldehyde is then converted to ethanol. So that's a further reaction from acetaldehyde to ethanol. And here too, you see that the coenzymes that are taking part in this reaction are NADH, NAD+. We've seen this. When you see this coenzyme, you know that you have oxidation reduction reactions occurring. Now take note of this enzyme, alcohol dehydrogenase, which is involved in the conversion of acetaldehyde to ethanol. There's something interesting we can talk about that practically. And that is, see, if you take a homologous series of alcohols, and what I mean by that is when you take alcohols in a series of the number of carbons that are in the structure of the alcohol. So if you take methanol, so you are looking at methanol here. Methanol has 1 carbon. The next ethanol will be the next alcohol will be ethanol, which has 2 carbons. The next will be propanol. Then you have butanol, have pentanol, hexanol. That's what I mean by homologous series of alcohols. Under normal circumstances, as the number of carbons increase, the toxicity of the alcohol increases. Or you can say the toxicity of alcohol increases with increase in molecular mass. Because once you have number of carbons increasing, you'll be having molecular mass increasing. And you know methanol is toxic. Because as you can see from this reaction pathway, methanol, when converted to, when it undergoes that metabolism under the activity of alcohol dehydrogenase, it forms formaldehyde. Formaldehyde is toxic. It's toxic to the central nervous system. It can result into even blindness, head turning, loss of consciousness. And you have that also going into the formation of formic acid. Formic acid is also toxic and result into tissue injury. But of course, this even the body has a mechanism of just breaking it down to give you carbon dioxide and water. And even you can have this going into the synthesis of folic acid. But you see, if this methanol is toxic in this way, then you expect that ethanol should be more toxic than this. That's what you expect. But interestingly, you saw, look at the reaction pathway. Let's go back to it. You see that alcohol dehydrogenase is acting on acetaldehyde. So if you take that into consideration, that here you have methanol getting converted to formaldehyde, what it means is if it is ethanol, it will be converted to acetaldehyde. And if it's converted to acetaldehyde, then alcohol dehydrogenase will act on that acetaldehyde to produce carbon dioxide, or no, to produce ethanol. So alcohol dehydrogenase will act on acetaldehyde to produce this way is further reduction, but if it was the opposite, in the opposite direction, where you have alcohol dehydrogenase acting on ethanol to give you acetaldehyde, the next thing you see is that that acetaldehyde is either oxidized now to give you pyruvate, and that pyruvate, you can have this process of decarboxylation, removal of the carbon dioxide from it, and then the acetic group can be conjugated to coenzyme A. So you have a formation of acetyl coenzyme A. And that acetyl coenzyme A can go into further metabolic processes. For instance, can go into the TCA cycle and generate energy. So that you find that ethanol is consumed by humans as an edible product. So you have production of BS, or even in some of our foods. If you put down corn dough for some time, under anaerobic condition, it starts to ferment. Kenke is a fermented product. It's an alcohol in it. It's ethanol. And it's not as toxic to us as methanol. because of the activity of alcohol dehydrogenase, converting it to acetaldehyde, and that acetaldehyde can get broken down into, say, decarbosylation will produce carbon dioxide, and then the acetic group will bind to coenzyme A to generate energy. So that is something interesting there, even though you refer to that homologous series when it comes to ethanol, it's not so for humans. But when you go ahead again, propanol becomes toxic. Butanol becomes more toxic, like that. And so you see, even when you make use of persons who get alcohol intoxication, let's say methanol intoxication, that you have a drug like formipizole, which is an inhibitor to the enzyme alcohol dehydrogenase. That is to prevent the conversion of methanol to formaldehyde. Because what it is that it is the metabolite of methanol that becomes so toxic to the body, so that if that enzyme which is involved in its conversion is inhibited, then you won't have the formaldehyde being formed. And the methanol can be excreted out of the body through urine. And this formipizole, in place of it, alcohol, the ethanol is used sometimes. So it's like alcohol intoxication is being treated with alcohol, because ethanol will also come and engage this enzyme. So it's a form of what you refer to as a competitive inhibition. Because once you have ethanol introduced into the body, alcohol dehydrogenase will preferably go to be acting on the ethanol and not the methanol. All right, so that is a practical application of that. Then we look at the process that occurs in bacteria. What we look at was in bacteria. Now we're looking at the process that can occur in animals and in humans, where we have lactic acid fermentation, what we refer to as lactic acidosis. Again, we take it from pyruvate. This time around, you see there's no decarboxylation. But what you see is that lactate dehydrogenase reduces that ketone group there. It becomes a hydroxy group. And that is lactic acid. Lactate, that's lactic acid. Now, this process, remember, is occurring under anaerobic condition. And what we refer to as lactic acidosis is what we commonly call muscle pull. So if you are involved in a vigorous activity and the amount of oxygen that is present in the cells to carry out this metabolic process of breaking down carbohydrate to generate energy for the cells is lower than what is needed, because you are making use of the energy so fast, so quickly, because of the vigorous activity you are involved in. Then what happens is that an anaerobic condition is created in the tissue. And in that instance, instead of pyruvate getting converted into acetyl-CoA and going to the TCA cycle to generate energy, it will rather be going into the production of lactic acid, which results into that muscle pull, what we refer to as the lactic acidosis. I mean, which is why, sportsmen, sportswomen are advised not to be consuming alcohol, no smoking, because you need your lungs, the performance of your lungs, to take in a lot of oxygen at once when you are involved in such activity. Generally, the lung performance of um, professional sports men and women is higher than the rest of us normal people, because we are not mostly doing things that will enhance our lung capacity. But if you do exercise continuously for some time, the lung capacity is a little bit increased, so that you'll be able to take in enough of oxygen that will go to the tissues, so that you have this process of breakdown of carbohydrates, generating enough energy for the tissues when you are involved in vigorous activity. Now we can take a look at, say, sucrose metabolism. These things are simple and straightforward. We look at it like in the reverse reaction, so that we see sucrose. So this is sucrose consisting of glucose and fructose. And you have a glycosidic bond between the glucose molecule and the fructose molecule. That glycosidic bond breaks down for us to get the monomers. It's a disaccharide, and when it's broken down, you have the monosaccharides, glucose and fructose. It's a simple reaction that you can easily relate to. So that's a breakdown there. Now, if you also take, say, fructose, because we've seen the reaction pathway that glucose will undergo, which we refer to as glycolysis. If you take fructose, you have what we refer to as fructolysis, where you have, first you have, again, some phosphorylation, and you have a fructose 1-phosphate. And that fructose 1-phosphate, you see, acted upon by an aldolase, that same enzyme that was involved in the lytic reaction in glycolysis. And the fructose 1-phosphate is broken down into glyceraldehyde. We saw this glyceraldehyde 3-phosphate. So from glyceraldehyde, you have a kinase, a triokinase, that is transferring a phosphate group from ATP onto it, and you have a glyceraldehyde 3-phosphate. So once you have the glyceraldehyde 3-phosphate, which is the end product of the phase 1 of glycolysis, obviously this will just undergo the second phase of glycolysis to generate energy. The second molecule is dihydroxyacetone phosphate. We saw also in the glycolytic pathway that the dihydroxyacetone phosphate gets converted to 3 glyceraldehyde 3-phosphate. There too is an isomerase, triose phosphate isomerase, and then that glyceraldehyde 3-phosphate goes into the second phase of glycolysis to generate energy. Now, if you have salivary amylase, we talk of salivary amylase breaking down starch. It breaks down starch into smaller molecules, because starch is a big molecule. Glucose units, a lot of them bonded together, and you have salivary amylase breaking them down into, say, this disaccharide, which is maltose. Maltose consists of 2 molecules of glucose. So 2 glucose monomers will form maltose. And it's also just a breakdown on that glycosidic bond, 1,4 glycosidic bond between them. Lactose is milk sugar, which is a disaccharide of galactose and glucose. And I think we've seen this enzyme, lactase, which hydrolyzes lactose into the monomers, galactose and glucose. Now, this enzyme, what we can say about it practically in us humans is that deficiency of this enzyme, lactase, in the small intestines, that is where you should have the breakdown of lactose into galactose and glucose. So if you have a deficiency of lactase, what then happens is that when you have lactose moving to the large intestines, you have digestive tracts microorganisms. So the microflora, the bacteria in the hindguts, will then be metabolizing these lactose molecules and be generating gas in some form of fermentation, which then will result into some stomach cramps, stomach rumbles. You see, well, when you're there, then the stomach is sort of making noise, sort of discomfort. In some people, this they can have some severe symptoms of, headaches and people have different ways that's like different symptoms of this. But one thing you should know is that this is not it's not an immune response. So it's not an allergy. It's not an allergy. It's just a deficiency of this enzyme that is resulting into the lactose molecules not being broken down, and so you have bacteria, in the digestive tract, rather acting on this, which will be producing a lot of gas, and that will result into discomfort and cramps and abdominal pains, things like that. All right, I think this should be the last thing. Like we have we've seen that the lactose is broken down into galactose and glucose. And we've already been clear about how glucose is broken down. How about the galactose? So the galactose is exchanged for glucose in this manner that you have glucose 1-phosphate, which is bonded on a nucleotide. That's a uridine monophosphate. So you are seeing another function of a nucleotide apart from its being a component of nucleic acids. You also see it here as involved in carbohydrate metabolism. It's like a carrier, just like the way we see transfer RNA carrying amino acids in the synthesis of proteins. You also see these nucleotides acting in this manner, where they are carriers of these monosaccharide units. Even when we come to the synthetic pathway, you see that in the formation of glycogen, for instance, you see this nucleotide involved in carrying and transferring glucose molecules one after the other to form glycosidic bonds in order to have a large glycogen structure formed. And here you see, if you look at the structure of galactose 1-phosphate and glucose 1-phosphate, there's only one difference in the structure. That is, on carbon number 4 for galactose, you have the hydroxyl group pointing upwards. In glucose, you have that hydroxyl group pointing downwards. So if you remember, galactose and glucose are epimers. That is, The only difference in their structure is on only one carbon, and that is carbon number 4. And so you see this uridine monophosphate carrying a glucose 1-phosphate, exchanges it for that galactose 1-phosphate. And whilst it is bonded, when you have that galactose 1-phosphate bonded onto the nucleotide, it is then converted into glucose 1-phosphate. So you see the hydroxyl group is now pointing downwards. So you have now, once you have that glucose 1-phosphate, of course, the glucose 1-phosphate can undergo isomerization reaction to give you glucose 6-phosphate, which then can go down the glycolytic pathway. So it's just a conversion. Think of it as a conversion of galactose to glucose, just by isomerization, the change of the direction of the hydroxyl group on carbon number 4. And that's all. And then it will get into the catabolic process as glucose. All right. So this is where I'll stop talking. And if you have any questions, any contributions, anything you like to say, you can write in the chat or you can raise your hand. Then I will unmute your microphone. I will enable your microphone. You unmute yourself and then you speak. The floor is open.